Hey, everybody. There's a new PowerPoint on Moodle, so go and snag that. Deals with reptile life histories, which is what we're going to be moving into today. We will not finish reptile life histories, I don't suspect. So I want to talk about the test on Friday a little bit. So normally I like to divide tests up into kind of logical segments, and I don't like splitting topics on a particular exam. So since we finish reptile life histories and reproduction, since we finished amphibian reproduction and life histories, we'll end the test material there. Even though we're going to start reptiles today, that won't be on the test on Friday. That will just be covered on the final. The other thing that I want to say about the final on Friday is that you'll pick it up in lab, which you already know about. The other thing that I want to say about that is that I was disappointed that people were using notes when I thought I made it clear that I didn't want people to be using notes. However, I'm also not a retaliatory type of person, so I'm not going to change my behavior on any of the future exams. The future exams will be exactly like they have been up to this point. And just to assure you that that is the case, while I normally make up exams from scratch every year, what I'm going to do for the next two exams is I'm going to essentially recycle exams from previous semesters. So I went back to 2017, which was the iteration of this course two versions ago, and I had trouble finding the exam actually, but I taught this course on iTunes U, and everything on iTunes U is backed up. So here is the old test, and unfortunately I don't have anything but the key has the original questions. So I am going to just give you a test from a previous semester that that covers the same exact material. The only difference is that the last two questions on the test deal with reptile life history and reproduction, and so I will pull those off of the test and create two new questions on the test that are focused on something from this test material section. But other than that, it will be a test from a previous semester, and you won't be able to say at the end of the semester, oh, Paul was mean to us after the whole test taking debacle of 2021. Paul is no meaner to you in this case than he was to students in 2017. So if that seems fair to you, let me know by giving me some sort of feedback. A thumbs up from Quinn. Okay, good. All right. Retaliation doesn't really gain anyone anything, so I don't know why people do it. It destroys trust, and it's just kind of stupid and petty. So I endeavor to not behave in those ways. All right. Lab this week on Wednesday is the last spring break day. I'm trying to find words. On that day, you won't have classes, so we won't meet on Wednesday, but we will meet on Thursday, and we'll go into the field just like last time. Hopefully it'll be nicer weather. It won't be rainy. The rain fogged up the camera lenses, which made it difficult to take photos. So hopefully the whole idea of taking photos will be a little easier. What I'm going to do with the photos from this past Thursday is hopefully sometime in the next day or two, I will pull those photos up. And what I will do is I will create folders for each of you on the Moodle site for any photos that were taken on my camera. And I'll just upload those photos onto Moodle for all the field trips that we take with one another, so that you have access to those. And I'll also get you a format for how I want you to turn those things in, including all of the collection data, et cetera, et cetera. So look for those folders. The only problem potentially with those folders is that there's an ultimate upload limit on Moodle, and I don't know if the photos will ultimately surpass that upload limit. So if that becomes a problem, then I'll figure out another way of getting them distributed to you. I know that photos that are of good quality in terms of resolution and pixelation and things like that often overload the email system's ability to transfer files that way. So I'll do something like set up a temporary Dropbox folder for you or something like that. You can then download the photos onto your desktop and then let me know when that happens, and then I'll delete them after that. So that's how we'll handle getting photos distributed. So this weekend lab, my plan is to go to Cooley Lake. So Cooley Lake is a conservation area, not far from here, actually. It's out towards Missouri City, east of Liberty, southeast of Liberty. And it's got these lakes that used to be a part of an old oxbow lake, and it's been dammed up, and I think they used to use it as fish hatchery. And then above that is a big bluff. And what we will do is we will start at the top of the bluff, and we will be searching the bluff primarily for reptiles. If we have time, we may drive around and go down to the wetland area, except that the wetlands are kind of deep, and it's best to go there with chest waders, which probably most of you don't possess. But there are also plenty of things to see down there in terms of flipping logs and whatnot. So once again, because this is Missouri Department of Conservation land, we won't be collecting anything per se, but we will have camera equipment and we'll be doing photography there. The following week, we will be going to the old golf course that is still owned by the college, and located, it seems like a golf course is a really stupid place to go and collect amphibians, reptiles, but there are a bunch of ephemeral pools that are on the outskirts of the golf course, and we will go and visit at least one of those, and we will collect some amphibians while we are there, and then we'll bring them back to the lab and preserve them, and that can be your preserved specimen for your collection. So that is that. Any questions about any of that? All right, I'm going to share my screen. This is my partner at Cooley Lake, at the bluff above Cooley Lake. We went there a couple of years ago. Among the specimens that we collected was this blue racer, Coluber constrictor. This is a photo of it. So when we think about the kinds of photos that I'm looking for you guys to produce, it would be nice if the photos were diagnostic, and that's one of the requirements, is that the photo be diagnostic. And so in this case, you can see that this is a long, slender snake. It has smooth scales, has a long tapering tail. The scales have this bluish tint to them. There's a very large eye on the head. You can't see any belly scoots, but you would see, if you could, that it's an animal with a slate plain white belly. So these are all characteristics that tell you that this is Coluber constrictor. The head is particularly distinctive in Colubra constrictor, especially when combined with the smooth scales. Those characteristics may not distinguish Colubra constrictor from other racers in other parts of its range, but those certainly work to separate this species from other snakes within Missouri. So the snake is fully in focus. It's well lit. You can see the distinguishing characteristics. And it's actually pleasant to look at. And so all of these things are things that I would like, your photos to have when you submit them as your specimen photos. Art is in the eye of the beholder, but it's better, even if it's in focus, it's better if the snake isn't actively trying to slither away from you when you snapped the photo. So these are just some examples of, even this would be a photo that would be easy enough for me to look at this photo and see that it is a blue racer from here in Missouri. I told you I'm on this Facebook group called Field Herpetology, and oftentimes people will submit photographs saying, saw the snake, I don't know what it is, and it's impossible for anyone to determine what it is because it's a photo that doesn't allow you to make any species level identification. So I definitely want the photos to be able to identify the specimen species. So whatever you have to do in order to do that, then do so. All right, questions about lab? All right, everybody can see my slides? Okay, all right. So learning goals for this section of material. We've already talked about temperature-dependent sex determination. We will revisit that. So we've talked about how it works, but we haven't talked about why it's an adaptive trait. So we'll be covering that information. So you should be capable of explaining that. Explain why climate change poses problems for species that exhibit temperature-dependent sex determination. Explain the mechanisms of parthenogenesis in reptiles and the taxonomic distribution of parthenogenesis. You should be able to explain the differences between viviparous and oviparous species and how these differences might explain the evolution of viviparity. Describe prenatal care in reptiles, the form of prenatal care, which groups tend to do it, which sexes tend to do it. Explain the pros and cons of having shelled eggs, and explain the constraints on reproductive anatomy that mode of fertilization places on reptiles. Be able to explain the basics of reptile embryology, which are somewhat different than the embryology and development of amphibians, which is one of the reasons that they placed reptiles in their own chapter in the textbook. And then once again, we're going to revisit life history theory and how it pertains to reptiles, although it pertains to reptiles in the same way that it pertains to amphibians. So this will be review to a certain degree. So that's hopefully what we'll get out of our treatment of chapter 9 in the textbook. So this is all review. This difference between environmental sex determination and genotypic sex determination. So remember that genotypic sex determination is where you have sex determined by the chromosomes. You can either have males or females that are heterogametic, and we've talked about in the past what heterogametic means. Humans have males being the heterogametic sex, meaning that they have two different chromosomes to make them males. However, in birds, for example, ZW are the chromosomes and the females are the heterogametic sex. So it varies taxonomically as to which sex is heterogametic. Temperature-dependent sex determination, on the other hand, is where sex is determined or influenced by the incubation temperature of the embryo. There's not a strict dichotomy, and there is still another spelling error on this, in spite of the fact that I thought I had gone through this already. So this isn't a strict dichotomy. For example, in the skink Bassiana duperei, males are XY, so they're a heterogametic species, but sex ratios are biased at low temperatures, and larger eggs produce females at a variety of temperatures. And so this is a situation where there is genotypic sex determination, but that genotype is then influenced by temperature and egg size in this case. Gecko japonicus is a TSD species where sex ratio is influenced secondarily by egg size at intermediate temperatures. And so once again, you can have genotypic sex determination that is influenced by temperature, and you can have a temperature-dependent sex determination situation that is affected by then another environmental factor, something like egg size. Egg size is oftentimes influenced by the amount of resources that a female has to put into eggs, and so egg size is influencing the nature of activity of temperature-dependent sex determination. So it's not a strict dichotomy. You're not just genotypic or just temperature-dependent sex determination. There are influences of both in a variety of species. These are just two examples. So we've talked about the mechanism of TSD. What does temperature influence in temperature-dependent sex determination?
The gene expression, which then inadvertently affects like the production of hormones, so like testosterone or estrogen, which then determines sex.
Okay, well, you went through the whole thing rather than me asking a series of questions.
Sorry, my bad.
That's fine. That's fine. So temperature affects the gene expression, and in specific, it affects the expression of the genes that produce reductase and aromatase, which are two enzymes. These enzymes are enzymes that both act on testosterone, but reductase basically converts testosterone into dihydroxytestosterone, which is the activated form of testosterone, and aromatase acts on testosterone to produce estrogen or estradiol. And then these two things act on the undifferentiated cells in the gonads to basically affect gonadal development, to turn those gonads into ovaries or testes, depending upon which hormone is being supplied. So the nature of temperature-dependent sex determination is that the temperature affects gene expression. That gene expression then affects the hormones, whereas in genotypic sex determination, those genes act without being influenced by temperature. If you're one sex, you produce you produce more reductase than aromatase, and you get dihydroxytestosterone. If you're the other genotypic sex, you produce estrogen. And so there is no temperature effect in genotypic sex determination. So we talked about these figures when we went through this earlier in the semester. Who can explain to me what's going on in these two figures? This is percent female and percent male. We have temperature and we have the concentration of estradiol, and then we have temperature and the dosage of something else. What's going on in these two figures?
So I think they were trying to manipulate sex determination. So I'm pretty sure for the figure on the left, females were produced at a higher temperature. And so you can see when they don't add estradiol in that first, I guess, row of temperatures, no females are produced at 26 degrees Celsius. And then the most females are produced at 29.2 degrees Celsius.
Yep. So where you have no estradiol being added, it just takes on the normal temperature-dependent sex determination pattern, which is females being produced at high temperatures, male-dominated at low temperatures. Yep. Continue.
And so as you add estradiol, even at low temperatures, you can see that it overcomes the amount of like male producing hormone that's being produced. And so as the more estradiol is introduced to those embryos, the percent female goes up even at low temperatures.
Yep. Kiara, take us through figure 6. Kiara, are you there?
Yeah, I'm looking at the figure.
Okay, all right. Keep in mind that they've reversed the order of temperatures here.
Yeah, so if you have an increased temperature, there's a higher percentage of males. And then if you have a lesser dosage, you're not going to have as many males.
And what are they dosing these eggs with?
That I don't know.
It's in the figure caption.
The inhibitor?
Yeah, aromatase inhibitor. So they're inhibiting aromatase. So if you inhibit aromatase, you're inhibiting the ability to produce estrogen. So if you're reducing the ability to produce estrogen as you increase the dosage of aromatase, what should you be getting more of?
I mean, if you're reducing estrogen, you're not going to have as many males.
Yeah. No, you're going to have, if you're reducing estrogen, you're going to end up having
More males.
more males. Okay. So normally at high temperatures, at 31 degrees, what do you get?
No males.
No males, but as you increase aromatase inhibitor,
You get more males.
decreasing the amount of estrogen being produced, you get more males, and that is pronounced at these not super high temperatures, but kind of on the high side, but more intermediate temperatures. You have some males being produced at high temperatures, which would normally produce females, but then as you add aromatase, you get more and more males. Aromatase inhibitor, you get more and more males being produced. And the reason that they did this experiment was they were essentially testing this model. In these temperature-dependent sex determination species, the idea is that gene expression is affecting hormone abundance. These hormones then, enzyme abundance. These enzymes then influence hormone abundance, which influences the differentiation of the gonads. And so you can overcome the effect of temperature-related gene expression by either inhibiting one type of enzyme or adding a particular hormone, which then once again indicates that it really is the production of these hormones that is regulated by enzyme activity, that is regulated by temperature-dependent gene expression of these two enzymes being produced. So that's that whole figure. You'll recall that we have different forms that this takes. In some cases, if we have percentage male on the y-axis and incubation temperatures on the x-axis, there are some situations where males are produced at cold temperatures, other situations where males are produced at warm temperatures, and then a third situation where males are produced at intermediate temperatures. And this varies by species. So we have Graptomys washitaensis, which is the Washita map turtle. And it is a situation where males are produced at cold temperatures, females are produced at warm temperatures, and there's this narrow range of temperatures over which the shift from male domination to female domination in the clutch occurs. In Pilomedusa subrufa, which is a type of side neck turtle or snake neck turtle, the females are produced more at really low temperatures and high temperatures, and males are produced at this kind of wide range of intermediate temperatures. And so this would be a type 1A situation with Graptomys washitaensis, And then this would be a type 2 situation with Pilomedusa. And so it just depends on the species as to which form this takes. So these two guys, Eric Sharnov and Jim Bull, back in the 80s, put forth this idea that the only way that you can have temperature-dependent sex determination evolving is if there is an adaptive significance to it. And the adaptive significance would be that your fitness as a male will be highest at those temperatures that produce males. And so in a situation like this, you would tend to have males being produced at intermediate temperatures because their fitness is highest at intermediate temperatures, and their fitness goes down at the extreme high and extreme low temperatures, whereas female fitness is lowest at intermediate temperatures and highest at extreme temperatures. This would lead to a situation where you would have males being produced at intermediate temperatures and females being produced at both high and low temperatures. So this type of fitness relationship would result in this type of sex determination mechanism. If you had a situation where males and females had different fitness surfaces, females are unaffected by temperature, whereas males experience higher fitness at intermediate temperatures. Once again, you might expect males to be produced more at intermediate temperatures, whereas females might not be influenced by temperature at all, but you would have more females being produced at the extreme temperatures simply because you have more males being produced at relative to one another in terms of sex ratio. You have more males being produced here, so it's a male bias sex ratio here, but you still have more females being produced than males here, so it's now a female bias sex ratio. So in all of these cases, these would be situations that favor temperature-dependent sex determination. However, this situation is not a situation that would favor temperature-dependent sex determination because temperature of incubation affects the fitness of males and females equally. And so Warner and Shine in 2008 tested this with a lizard in Australia, and they found that the incubation temperature was correlated with the fitness of the male, and the incubation temperature was also roughly correlated with the fitness of the female in a situation that resembles panel A up here. And so what Sharnov and Bull basically said was, in order for temperature-dependent sex determination to evolve, there has to be a situation where the incubation temperature of males and females is correlated in terms of their fitness consequences, but those fitness consequences have to be different for males and females at the different temperatures. And the tricky part of that has been finding experimental evidence that actually shows that this is the case. And the reason for that is it's very hard in a lab to produce males and females in enough numbers at those different ratios to test whether or not temperature actually influences fitness. So take an example where you have a turtle species that produces, if this is percent male, you have a turtle species that does this. If this is the incubation temperature, and you have this relatively narrow range of temperatures that produces males, well, you could incubate eggs down here at 27, and you could incubate eggs here up at 32. That would give you high temperature females and low temperature females, and you could incubate temperatures at 29.5 degrees, where you would get 100% males. But what you don't have in any of these situations is you don't have any males at high temperatures, and you don't have any females at intermediate temperatures. And in order to test this model, you need a mixture of both males and females. So you would need this curve to be something more like this, where you produce both males and females at this intermediate temperature, and you would need to have this whole thing being kind of just flattened out so that you have some males being produced at temperatures that normally produce females. And it turns out that's really difficult to do in a lab setting. And then once you've produced those animals, you have to then have a way of measuring what the fitness is of those different individuals. So imagine that you do produce low temperature males and females, intermediate temperature males and females, and high temperature males and females. You then have to put them into an environment that allows you to look at what the fitness consequences are of those things. Because many of these species are things like turtles, which are exceptionally long-lived, it's difficult to then measure fitness consequences. So there are a number of kind of practical challenges with doing this kind of work, which is why some people will actually, like what they did in this particular study with Warner and Shine, they didn't attempt to do this with temperature. They incubated eggs at different temperatures, and then they did these types of aromatase inhibitor and estradiol additions to produce males and females at temperatures where they normally wouldn't be producing males and females. And so they incubated the eggs at a constant temperature, manipulated sex using either hormone additions or enzyme inhibitors to produce males and females at temperatures that they would normally not be produced at. And then they put these in these open exclosures out in nature and then look to see what the fitness consequences were. And in this case, the fitness consequences were number of offspring. So it's the most direct fitness measure that you can imagine. With turtles, people often do this with 1 or 2 year survivorship in the wild using mark and recapture methods. Now that we know how to manipulate the hormones of eggs being incubated at different temperatures, it's a little easier to do these types of experiments than it was back in the 1990s when a lab mate of mine was trying to do this with snapping turtles. But still, there's also the complication of measuring fitness after these animals are hatched. So lots of practical challenges with actually measuring this. As it turns out, the distribution of sex-determining mechanisms is very scattered across the phylogeny of reptiles. So if we look at all of these reptile groups, we see the red ones are independent evolutionary developments, convergent evolutionary events of temperature-dependent sex determination. So we have temperature-dependent sex determination in a group of geckos. We have both genotypic sex determination and temperature-dependent sex determination also in a number of geckos. But one of the things that you'll notice is that the sex-determining mechanism has evolved repeatedly over the course of the evolution of the reptiles. Many different independent evolutionary events that produced TSD. And the other thing that you'll notice in this phylogeny is it not only tells you in those cases where you do have temperature-dependent sex determination, it also tells you what types of temperature-dependent sex determination you have. So even within Crocodilia, you have 2 different types of sex determination. One type of sex determination is 1B, which is this one, where males dominate at warm temperature, and the other one is type 2, where males dominate at intermediate temperatures. So even within a taxonomic group, the mechanism of sex determination in temperature-dependent sex determination is not consistent. And so a lot of evolutionary flexibility in this particular characteristic. So when we think about temperature-dependent sex determination and climate change, it's easy to imagine what might be happening with organisms out in the field. And there's this paper published back in 1994 that surveyed naturally occurring nests of Caretta caretta, which is one of the sea turtle species. And they found that 94% of hatchlings of Caretta caretta are females. So if we go back to this figure, in either 1A or 1B, as temperatures increase, as environmental temperatures increase, you risk for any given species ending up with a climate full of microhabitats that are only suitable for producing one particular sex. So if you're a 1A TSD species, as the climate warms, you're going to produce only females. If you're a 1B, you will produce only males. If you are a type 2 species, what happens is you don't get any females being produced at the low temperatures, but you get males and females potentially being produced, but perhaps altering the sex ratio. And if it gets warm enough, you might phase out males altogether and once again get only females, because the latter half of this figure 2 here, looks just like 1A, just shifted upward in temperature. So the conservation consequences of this is that you could end up with species that only produce one sex or the other. And that's bad if you're not a parthenogenic species, which most of these species are not. In some cases, organisms can alter their behavior, but in many cases, they may not have behavior that is flexible enough to cope with the lack of available microhabitats for egg laying. And in this case, you might just end up with a population full of females. One of the arguments for why this system might have evolved is that over the course of especially long-lived organisms, there may be hot years, there may be cold years, but when it comes down to it, if there's an even number of hot years and cold years, you get an even mixture of both males and females. And so this allows organisms to be somewhat flexible as climate changes. But that mainly deals with year-to-year variability rather than a directional trend of climate change. Questions about temperature-dependent sex determination? You will notice in this figure that sex determination, temperature-dependent sex determination, tends to be found in species that are relatively long-lived. So you see it in the crocodiles, you see it in all of the turtles, you see it in some of the skink species. These also tend to be larger, longer-lived skinks that exhibit this, and in some of the geckos, which can also be relatively long-lived, the ground geckos in particular, which tend to be larger and more longer-lived than some of the other gecko families. And so, not surprising that temperature-dependent sex determination would be found in longer-lived species. And then we come to asexual reproduction. And we've talked about this a little bit in the past also. This is especially prominent in the family Teidae, the race runners. And once again, we see that this is one example where we have hybridization going on between Aspidoscelis tigris and Aspidoscelis inornata. And when these two species interbreed, they produce a hybrid parthenogenic form, Aspidoscelis neomexicana. And this species then represents its own evolutionary species because it is not capable of exchanging gametes with either of the parental species. And so normally under regular meiosis, what happens is you start off with a single diploid cell. This is during gamete formation. You get those chromosomes replicating during the S phase of the cell cycle. And then as you move into meiosis, at the beginning of meiosis in prophase 1, you get synapses occurring. Synapses leads to an exchange of chromosomal ends, such that when you reduce the number of chromosomes in a cell, you end up with just one of the homologous pairs, and then the sister chromatids separate in the second phase of meiosis, and you get haploid offspring, or haploid gametes that then become sperm or egg, depending on which sex you're dealing with. In the case of asexually reproducing species, you end up with replication happening back before the S phase of the cell cycle. And so you get a doubling of the chromosomes, so that during the S phase, rather than just adding a sister chromatid to a single chromosome, you actually duplicate the chromosomes. And so you end up with a diploid organism that then produces a tetraploid cell, and it's that tetraploid cell that moves into the process of meiosis. And so this is what we refer to as endoreplication prior to meiosis. And so in your book, they refer to it as meiotic endoreplication. And this is what enables parthenogenesis, because then the animal goes through the process of meiosis and ends up with a diploid gamete at the end. And this diploid gamete at the end then develops into a new individual. And so the nature of parthenogenesis is that you produce a new organism without the benefit of an extra gamete from a partner of the other sex. Now, in both of these paths, you get production of diploid gametes, and these diploid gametes can be used to produce new offspring. But depending upon how the homologous chromosomes line up, you either get increases in genetic diversity, or you basically get clones. In this case, the female starts off with one blue chromosome and one red chromosome. This endoreplication then just doubles those things. Then during the S phase, you get replication occurring. But in this case, the sister chromosomes get paired up during prophase 1 of meiosis. And because of that, even if crossing over occurs, it's crossing over between homologous pairs. And so there's no generation of new genetic variation. You go through the first meiotic division, it divides the homologous pairs into two different cells. And then those homologous pairs split the sister chromatids, those two chromosomes split the sister chromatids, and you end up with a group of offspring that are all genetically identical to one another, and they're also all genetically identical to the parent. Other forms of parthenogenesis, however, the homologs pair with one another rather than just the sister chromosomes pairing with one another. You still get crossing over occurring. You get the exchanges of these end segments of the chromosomes, which means that after the first meiotic division, you end up with 4 sister chromatids. Each of those sister chromatids is genetically different. So when you then divide those during the second phase of meiosis, you end up with offspring that are still diploid, but in this case, all of the offspring are slightly genetically modified from the genotype of the parent. So you can have parthenogenesis working one of two ways, one way which produces clones of the parent, one way which produces genetic diversity in the offspring that are different from the parents. But in both cases, these are parthenogenic mechanisms of producing gametes. If I recall correctly, and I have to go back and check myself on this, in most Tia lizards, you end up with this type, the sister chromosome pairing. And so you end up with actually clones of the parents, of the parent species, in this case, the parent female. So this obviously only occurs in species where all of the individuals in the species are female. That was a lot of information delivered in a single slide. Do you have questions about that? So pre-meiotic endoreplication refers to this replication of the chromatids before you enter the process of meiosis. So in some
I have
cases,
a quick,
yeah, go ahead.
So in, I think it's called kleptogenesis in the salamanders, when they, I know it rejects the male genome. Is it, I don't understand how though. Like, because I imagine it happening like this versus.
Yeah, so we know a lot about this from simply sequencing things, and the cellular mechanisms by which the unused genome gets rejected are really unclear. And so what we do know from testing them, so you can take, and this has to do with the next slide coming up here in a minute. You have these all-female Ambystoma species, and they will tend to mate with the parent species that they were hybridized from, and they receive the sperm from those parent species, but those sperm never actually become incorporated into the developing new offspring. And so the parent species is adding sperm to the female that is the parthenogenic species, but that parental genome never becomes incorporated into the developing offspring. And they're not sure exactly why that genome doesn't become integrated, but when you genetically test them, you see that genome is not present in the offspring, in spite of the fact that the females mated with males of one of the two parent species. And so in many cases, in order to stimulate the development of the diploid eggs in parthenogenic species, you actually have to have copulation. And in the case of those kleptogenetic salamanders in the Great Lakes region, those ambystomatids, copulation includes the transfer of sperm, and then that sperm just doesn't become incorporated into the offspring. But in some cases, it's just the act of copulating that enables the process of parthenogenic production of offspring to occur. And so in the case of Tiid lizards, it's somewhat different. In Tiid lizards, the individual female lizards will actually copulate with one another. One of the lizards will kind of act as a female, one of them will act as a male. And even though the female doesn't have hemipenes, the physical act of, they refer to this as pseudocopulation, actually prompts the female's diploid eggs to then shift out of being static into starting the process of cell division. In some cases, it's the females of the species that interact with one another. In other cases, you will have the parthenogenic species once again copulating with males of the parent species. And once again, that act of copulation doesn't do anything in terms of transferring sperm to the eggs. It just is the environmental stimulus to prompt the division of the eggs. And it just varies by species as to what happens within the Tiids. But with the ambystomatids, cellularly, what is going on, nobody really knows yet. Unanswered question. So this is just a drawing from your book showing two female parthenogenic lizards involved in pseudocopulation. Although this is one of the weirder artist depictions of something going on, because it looks like the one female has grasped this female by the side of the body in such a way that it looks like it has embedded its lower jaw inside the body, rather than like just grabbing a skin fold or something. It's one of the more disturbing artist renditions that I've seen. You will oftentimes see with lizards copulating that the male lizard in species where there's males and females, oftentimes the male will hold down the female by biting the nape of the neck, biting the back, sometimes biting the hind legs. And then there's always the presence of hemipenes, which we'll talk about the structure of hemipenes later. Hemipenes located at the back of the male, and those are the intermittent organs that they use to get sperm inside the female. And hemipenes are their own scary sexually dimorphic sex organ. So, other questions about asexual reproduction? All right, we can think of reproductive modes in much the way we think of reproductive modes in the amphibians. We have lesotropy versus placentotropy or matotropy. Just refers to how the developing embryos are being nourished. Lesotropy are eggs where the yolk is doing all of the supplying of nutrients and energy to the developing embryo, whereas matotropy and placentotropy are being provided by the female through either the placenta or by some sort of substance that the female produces. So placental reptiles represent, once again, a continuum between these two extremes, but they all give live birth, and so your book refers to all of them as viviparous, and so this becomes this dichotomy between oviparity and viviparity. Much in the same way that we talked about what this continuum is in amphibians, a lot of the difference between oviparity and viviparity have to do with how long eggs are retained in the female. The longer eggs are retained in the female, the more embryonic development happens inside the female, and you're moving away from oviparity towards viviparity on this continuum. So there's not a lot of new material in terms of thinking about reproductive modes. One of the main differences between reptiles and amphibians is that the shell in this case, the egg in this case is shelled, and it has either a parchment shelled, a leathery shell around the egg, or an actual calcareous hard shell around the egg. Most reptiles lay parchment shelled eggs. A fewer number of reptile groups lay hard shelled eggs. And then in the course of doing this, because they're laying these eggs on land, oftentimes these eggs are laid in protective spaces that just over time have become called nests, even though they may not resemble nests as you think of, for example, a bird nest, but they're still nesting species to keep the eggs in one spot and to keep them protected to a certain degree. So the main thing that, the main differences that separate kind of what we refer to as parchment shelled eggs versus hard shelled eggs is just the proportion of the eggshell that is made up of these impermeable mineral layers. And so on the outside of all eggs, there is a shell membrane. And then in the shell membrane, what is added to the outside of the shell membrane are either layers of connective tissue, things like collagen and keratin and things like this, or the production of actual mineral layers on the outside. So turtles can either lay parchment shelled eggs or rigid shelled eggs. And what really determines this is the extent to which the mineral layer is built up. The thicker that layer, the more rigid that egg will be, simply because as you build up this layer of minerals, those layers become more and more tightly packed with one another. Whereas when the layers are short, there are these big spaces between them, which allow the egg to continue to be flexible to a certain degree. Once again, crocodilians have relatively thin mineral layers relative to the rest of the egg. Same thing with the tuatara. And then in the case of most lizards, there are no mineral deposits at all on the outside. And as a result, these what your book refers to as shell units aren't present. and then that leads to the most flexible sorts of shells. There are some lizard species that do lay rigid eggs. One of these is the geckos, the true geckos in the family Geckonidae. And as a result, these things also have shell units, but they're not as well defined as they are in the other groups. They just basically make this solid mass of shell that surrounds the membrane. Obviously, the presence of the mineral shell, what does this do to the properties of the egg that have enabled reptiles to escape water during reproduction? Escape this limitation of needing to lay eggs in water. Quinn, what does this impart to reptile eggs that enables them to escape water for reproduction? Is Quinn with us?
Yes, I'm here. I'm
She
just
is with us.
unsure of the answer.
Okay, as you put more of this mineral content on the outside of the shell, that increases the thickness of the shell. If you're going to leave the water for reproduction and start reproducing on land, what's the main environmental constraint that you have to get over, the environmental challenge that you have to get over? You don't want your shells to do what when you lay them on land?
Break.
So you don't want them to break, and so that would be one reason for the mineral layers on the outside of the shell. We actually didn't talk about structural support when we talked about reproduction in water. One of the things that you don't have to worry about when your eggs are in water is they don't have to retain their shape. They can actually take on any form because they're supported by the buoyancy provided by the water. But that's not what I was thinking of, although that's not a bad answer. What other big thing as you move from water to land do you have to think about from the standpoint of a nice moist egg?
The presence of a shell prevents desiccation.
Yes, the presence of a shell prevents desiccation, and the thicker that shell is, the more desiccation is going to be presented. So this actually brings up an important point, Quinn. On the last exam that you guys took, there was a question that I asked, and I don't remember what question it was, but a couple of you answered with an answer that I was not expecting. And I made a note on your paper, oh, that wasn't the answer that I was expecting, but that's a perfectly valid answer. Simply because it wasn't the answer that I had in my head, it doesn't make it wrong, it just makes it different. In the same way, if I had asked you about this on the test, and Quinn, if you had said, well, it keeps them from breaking, I would have looked at that and say, oh, that wasn't the answer I was going for. The answer I was going for was desiccation, but breakage is certainly a reasonable reason, or a reasonable advantage of having a shelled egg. If you're going to lay it on land, there's no water supporting the egg, and so in order for the egg to retain its shape and not just kind of flatten out into a pancake, having that added structure around the egg helps it maintain its shape, helps it from becoming damaged, and I would grade that as correct, even though it wasn't necessarily the answer that I was looking for. Now, in that answer, you would have had to have mentioned things about the buoyancy of the egg provided by water. Water pressing it on the egg helps the egg maintain its shape and maintain its integrity and things like that. But if you had done that, I would have been like, well, okay, that's not the answer I was looking for, but that's a reasonable, good answer. So correct. So when you look at the key to exams and things like this, note that even though you might not have had the answer that I had on the key, but you got credit for it, is because even though that's not the answer that I provided, it means that you provided an answer that was equally valid in the process of thinking through it. This is actually one of the challenges of writing a test, is you oftentimes write a question with a particular answer in mind, and you try and craft the question such that it leads to only that answer, but you didn't foresee answers that you weren't thinking about at the moment that you were writing the question. So anyway, well, we still have some things to talk about in terms of egg structure and the transition from oviparity to viviparity, but we'll just start here on Monday, which seems like forever from now, but that is the way it is. Questions? All right, I will see you guys on Thursday, and let me talk to Rose Galletti for a minute, and I'll see the rest of you on Thursday.
Hello.
Hey, Rose, did you come in on the weekend at all?
No, I ended up having to clear out my dad's apartment.
Ah, stinker.
But
Okay.
I honestly think I'll be fine because I have photos of all the specimens. So I
Okay.
feel like it'll be fine.
Okay. Hold on, let me turn off the